Thanks for joining IIIP 2020 virtual meeting. I'm Chang Lee. I'm the MAM student of Poultry Diagnostic and Research Center at UGA. Today I will talk about histological evaluation of the effect of high incubation temperature on embryonic development of thymus in broiler chickens. Temperature control has the biggest impact on the embryo development. Factors that can be impacted included hatch performance and chick quality. What we see on the screen in the machine, that is external air temperature. The temperature inside of the egg the embryo actually experienced is known as incubation temperature. However, measuring the internal egg temperature is destructive. Because of this, the egg shell temperature is widely used in many places, is non-invasive, and indicates the internal egg temperature closely. Medical infrared thermometer is commonly used for the measuring actual temperature because of its accuracy and convenience. However, when the machine door is opened, the temperature can fluctuate because of its influence of outside temperature. Because of this shortcoming, using a logo-type thermometer that is attached to the egg-shell or using installed temperature sensor in the setter trays are suggested alternatives. To maintain actual temperature at 37.8 Celsius or 100 Fahrenheit during whole incubation provide the best result of hatchability and chick quality. During incubation, embryo itself produces metabolic heat as a result of nutrient utilization. First week of incubation, the embryo produces very little amount of heat, but at the last week, it produces larger amount of heat as it gets bigger. Because of this, excess heat accumulated inside the incubator needed to be removed. To accomplish this, the incubator temperature needed to be adjusted as seen in the second chart. However, some type of incubator, especially multi-stage system, may not easy to adjust temperature. Then, at the late period of incubation, the elevated actual temperatures are experienced. High actual temperature in the late period of incubation resulted in adverse outcomes such as lower hatch performance, decreased chick quality, and immune suppression. There is a programmed embryonic development of hematopoietic system. The formation of bursa occurs in the middle of incubation. The hematopoietic cells colonize in the bursa mesenchyma at 8 to 15 days of incubation, whereas thymus development has 3 distinct waves. First wave is contributed by para-aortic foci around 7 to 8 days, and second and third waves originate from bone marrow. Last wave of colonization occurs around 17 to 20 days of incubation. So, hypothesis could be that the high incubation temperature in late period could have a significant impact on thymus development. The purpose of this study is to understand the effect of high incubation temperature in late period of incubation on thymus development through histological assessment of thymus by using ImageJ program. Therefore, the practical indicator of immune suppression during incubation can be established. 2 groups of eggs were set in an identical tabletop incubators. Incubation temperature for control group measured by actual temperature were maintained at 100 Fahrenheit. The heat-treated group temperature was increased by 2 degrees after 15 days of the transfer. After transfer, try to maintain a similar condition for both groups and kept them near 100 Fahrenheit or slightly lower than that. After hatching, all chicks were bulk weighted to calculate average chick weight and yield. Chick length was also an important measurement taken during this procedure. We randomly selected 50 chicks for yolk-free body mass and bursa weight. 40 birds has their thymus taken for histology, and 10 has a blood sample taken for cytology. Actual temperature was monitored by TinyTech temperature loggers. TinyTech has an external proof they can detect fast-changing temperature in difficult-to-access area with 0.01 Celsius sensitivity. 2 loggers are positioned for 1 incubation group. 1 logger has a monitor installed, and it helps in adjusting incubator temperature without opening door. Actual temperature data indicate that the control group was maintained 100 Fahrenheit within 1 degree of variation. The heat-treated group started at 101 Fahrenheit on the first and second day, then it maintained close 100 Fahrenheit until it was elevated by 2 degrees at 15 days of incubation. Summary of hatch performance. A total of 162 eggs were assigned to each incubator. Before setting eggs, individual egg weight was measured and graded by 5-gram difference. Then evenly divided 2 groups to make similar egg weight distribution to minimize the interference of heat production by different size of egg. True infertility was very low as indicated by the flock age, which was 40 weeks old prime flock. Average weight loss was 11.5% and chick yield was 70%, which was in optimal range. However, there is an accidental temperature drop after transfer for several hours. Due to this, the hatch window was extended around half day and resulted in draggy hatch and hatchability loss. Hatchability was 64.8% in control group and 66% in the heat-treated group. It was observed that the heat-treated group started hatching several hours earlier than control group. This study showed that the high incubation temperature influenced on embryonic development, especially in chick length and yolk-free body mass. These findings are consistent with the previous research. Body weight is not a significant indicator looking at this result, but yolk-free body mass is clearly an important indicator for showing different metabolic efficiency during incubation. There is no significant difference in bursa weight here. Looking at the distribution of chick length, there are more chicks with a shorter chick length position in the heat-treated group, which could indicate a chick quality issue. When birds were exposed to stressor, the population of lymphocytes decreased and heteropyle increased. Therefore, the ratio of heteropyle to lymphocyte used as an indicator of stress. Prior researchers reported that normal HL ratio was 0.38 in the older chicken and 0.9 at day-old chick. However, in this experiment, mean HL ratio was not significantly different, and it was higher in both groups in comparison to previous research. This means that both groups experienced a significant level of stress during incubation. The temperature drop after transfer and extended hatch window may play a role as additional stress. Thymus is spin-shaped and lies parallel to the vagus nerve and internal jugular vein in the neck. Chickens are known to have separate 7 lobes in each side. At day-old chick, thymus is embedded into adipose tissue, so each lobes are not easily distinguishable, and around 5 to 6 lobes were counted. In the microscope, lobes were irregularly folded and expanded with multiple lobules like grape shape. Number of lobes counted as 6 to 7. To measure the thymus parenchyma and cortex size, the digital photograph of histoslide at 1.x was taken. 2 representative lobes from one bird were picked and processed by ImageJ program. Once the representative lobe were selected in digital photograph, the color threshold function of ImageJ allowed to select total parenchyma as well as the cortex area. This is distinct from medulla. The range of color and its saturation are easily adjustable depending on the cellular density and the condition of staining. Area is measured by pixel, and the ratio was calculated by Excel program. ImageJ program is convenient when selecting the cortex area by using the color threshold function, even though it possesses a complicated structure. There is different degree of medulla development in thymus. A smaller sized thymus lobe has not established a medulla structure yet, so its cortex to parenchyma ratio was higher. In contrast, large and well-developed thymus lobe has mature cellular matrix, which form the medulla in its center and cortex in subcapsular area. As a result, its CP ratio was lower. There is a significant difference in size of parenchyma, however, it was observed to be larger in the heat-treated group. It is difficult to determine whether control group was suppressed in development or heat-treated group was advanced. There is no significant difference in CP ratio in either group. There is a negative relationship between parenchyma size and CP ratio. A bigger size of lobe tend to have a lower CP ratio. However, Bottom line indicate that, regardless of size of thymus, CP ratio was not less than 0.45. This number could suggest the maximum level of maturation of thymus at hatch. From this study, we found that elevated incubation temperature at 15 to 18 days of incubation negatively affect embryo development and chick quality in terms of yolk free body mass and chick length, which is consistent with the previous study. However, HL ratio was high in both groups. This means that after transfer, there are another stress factor present in both groups. Thymus development was influenced by the condition of late incubation. However, there could have been an additional factor as the heat-treated group possesses bigger thymus than control. Thymus CP ratio measured by image J represents the degree of medulla development at hatch. This morphometric assessment provides a potential chick quality indication to reveal the immune status in a quantitative way. Also, collecting data from the commercial hatchery will add value for this finding. Further study requires to investigate the effect of high incubation temperature extended after transfer. Here are the list of references for this study. I would like to address special thanks to who provide tremendous help for this project. Our MAM clinician and my colleague Katie and Linnea, Dr. Williams and Dr. Jordan at PDRC, Dr. Fred Hare at Veterinary Diagnostic Pathology, Dr. Empai Nansei at ABSN, and Dr. Louis Burgos at UGA. Thanks for your attention.